All right, sounds good. So I'll start here. Is everyone seeing my slides here all right?
Looking good, yes.
Yeah, perfect. So, all right, so let me just give you some background on this research that I've been doing for the past 6 years now, actually. So John and Uli have already sort of given a little bit of a background, but generally speaking here, the idea is we're trying to track some mechanisms of learning and value-based decision-making. And in neuroscience, generally speaking, there have been computational theories of how decision-making is done, suggesting how these problems might be solved by organisms. And one thing that's really important is in these theories, in these computational theories, certain variables are being kept track of by models. And generally speaking, we want to correlate some of these variables of interest to brain activity. So that's in very broad words, the theme of my research. So let me give 1 or 2 slides of background here so we can all be on the same page. So first of all, I just want to introduce this because this is the theoretical foundation to our work. So basically in machine learning, there's this class of algorithms here that can be used to maximize rewards from the decisions that we make in some environment, right? And this theory is named reinforcement learning. These algorithms are typically used by reading the state of the environment and then learning which decisions you can make in that environment to maximize your rewards in the long run. I'm including here some examples of applications for this, such as learning how to play Atari games, often at superhuman level, or using continuous control to teach an agent how to walk from scratch, or even learning how to play from scratch in an extremely complex state space, such as in the game of Go, like this, again, at superhuman levels. Now, given the success of reinforcement learning theory in explaining learning in general, in neuroscience, we have tried to take these models and adapt them to explain some form of neural activity during learning, right? And how decision-making happens. So I'm showing here just a few examples which are relevant in the context of this talk, but there are many more, of course. For instance, like in this example here, the spike rates, the rate of activation of dopamine neurons in monkeys, for example, they reflect the degree of surprise that you experience when you receive a reward. And subsequently, it can also express the amount of surprise that you get when you observe a stimulus that itself predicts the reward. So this surprise value is the same value that is used by as a learning signal. Reinforcement learning is that I'm talking about. So the same surprise value that acts as a learning signal was then later found to correlate with activity in the human ventral striatum during Pavlovian conditioning. So this is one example here that I'm showing in fMRI. And additionally, one example that is going to be really relevant for the context of my talk is this third one here, which is the fact that if you assign values using reinforcement learning to perform certain actions, let's say by performing an action with your hands, then these values also correlate to brain activity in certain areas. And specifically here, I'm going to be interested in the activity that occurs in this area called supplementary motor area. In the case of my study specifically, the pre-supplementary motor area. So given all of this, what we wanted to do was take advantage of this unique opportunity that we had to study learning processes using a very high level of temporal and spatial resolution that was afforded to us by invasive recordings in the human brain. So this was only possible because we had this opportunity to work with epilepsy patients at the Cedars-Sinai Medical Center in Los Angeles. These patients, as I'm showing here in the setup, they're implanted with intracranial microwires only for epilepsy monitoring purposes. And then while they're there being monitored, that's where I come in with my games, with my tasks. And then we can record from these patients signals like this one here. So we have a raw trace in which you can already see some neural spikes. And then we pre-process the signal. There's an entire pipeline to do this, which I'm not going to go into detail. But in the end, we extract action potentials. We can get to the level of single neurons. So with this data and with this motivation, I just want to share here the similar logic that all of our studies have. So I'm going to summarize it here so we can all be on the same page. So I only have to introduce this once. So let's say we have patients here that are performing some learning task, right? And then from performing this task, patients will produce both behavioral data, which could be, for example, making decisions, as well as neural recordings, such as this raw trace that I'm showing here. Then eventually, behavior will inform the creation of some reinforcement learning model that usually selected in order to reflect the best fits to behavior. Then finally, we try to correlate the neural data that we collected to some variables of interest, including those that we extracted from the models. So this is in very broad lines, the whole pipeline for the work that we do. So every single study that I talk from now on will have this same structure. So what do I want to talk about? So I want to discuss some specific aspects of how people encode information about values and how they use this information to make decisions. So I'm going to divide my talk in 3 parts, which correspond each one of them to our 3 main studies. In the first part, I'm going to talk about what happens when people make decisions under uncertainty while exploring uncertain environments, such as gambling with slot machines. In the second part, I'm going to take a step back and discuss a little bit the construction of cognitive maps during Pavlovian conditioning, which is, you could argue it's a simpler form of learning, but there's still a lot of rich behavior that we can extract there. And we study this by giving candy to patients during a learning task. So I'm going to talk about it, how we did that. And in the third part, I'm going to discuss observational learning, which we studied by having patients learn either by playing a game or by watching somebody else play a game. And then we compare those 2 things. So that's what I'm going to talk about. I'm going to give right away going to give all of you right away the key takeaways from my talk, just everything summarized, and then I hope that I'll be able to convince you of these things by the end of my talk. So first of all, we found that neurons in the human pre-supplementary motor area integrated components of value during decision-making under uncertainty, including expected values, uncertainty, and novelty. And subsequently, neurons in the same brain area also encoded the patient's decisions that they made during the task. Second, the second result that I'm going to talk about is how neurons in the human ventromedial prefrontal cortex represent a few important variables that we think are fundamental in the construction of cognitive maps during Pavlovian conditioning. So that's going to be the second key result that I'm going to discuss. And the third thing is we found that the human amygdala encoded values as predicted by reinforcement learning during observational learning and distinguished self-experience learning from observational learning. So this is going to be the third key result that I'm going to discuss here. So let's get into part 1. And I'm going to talk a lot more now about making decisions under uncertainty. So just to illustrate the problem here, let's imagine here, just for the sake of the argument, that you're trying to decide where to get dinner on a given night, okay? This is just to illustrate the problem. Maybe you open your favorite food app and you see there are 2 options. Let's say a pizza restaurant that you visited 10 times in the past and a taco restaurant which you have only visited once. Maybe you tried many things in the pizza restaurant before. Maybe you have even a pretty good idea of what the level of service is going to be there. In the taco restaurant, however, you probably have many options in the restaurant there in the menu that you haven't sampled from yet. So you don't have as good of an idea of what the level of service is going to be there. So in other words, even though you may like the pizza place, maybe there's some unexplored option in the Mexican restaurant, which you would enjoy even more. This is in essence the dilemma that we call the explore-exploit dilemma. Do you explore the taco restaurant out of curiosity, or do you exploit the information that you collected in the past about the pizza restaurant? So the key underlying variable in this mental computation is uncertainty. So here I'm going to define uncertainty. By the way, we have a very precise mathematical definition, but I'm just going to define it in broad terms here. Uncertainty here is being defined as your knowledge of the possible, the distribution of the possible outcomes that you can get from choosing some of the options. So the number of times you've been to the restaurant will act to reduce uncertainty because you get better at predicting the outcome value of your action of going to the restaurant. In theories of exploration, optimal theories usually, or optimal algorithms, I should say, they usually focus on strategies that target uncertainty reduction specifically. So that's one thing we should keep in mind right now. Now, there's a second variable that I want to talk about, which is novelty. So let's talk about it a little bit and why it's different from uncertainty. So let's say that one day you open your app again, and there's an entirely new option, which you haven't seen before. So not only you haven't been to the restaurant to sample from it, but you didn't even know the restaurant existed, right? So one variable here that might influence your decision now is novelty. So previous behavioral psychology studies have argued for the importance of novelty in decision-making, and we are considering this variable separately from uncertainty since it could be processed differently by the brain. In our study, we have a very precise mathematical definition for how we're using novelty. I'm going to define it with broad strokes here. We're defining novelty as the number of times, as a function, I should say, of the number of times that you saw that option as one of the options that was available to you, regardless of how many times you actually sampled that option. While uncertainty and novelty are typically correlated, they should be, of course, because a novel restaurant is also maximally uncertain, right? So they should be correlated, but conceptually they're distinct, and therefore they might be encoded differently by the brain. Our experimental design that I'm going to talk about here will be done in such a way to disentangle the influence of these two variables on behavior. Additionally, one last variable that I'm going to talk about here is expected values. So this can drive your decisions about restaurants because let's say you've had a good meal many times at the pizza restaurant, then every time you have a good meal in a restaurant drives up the expected value that you have there. Conversely, if you have bad experiences in a restaurant, the expected value of the meal that you're getting there in the future will go down. So if you take all of these three variables together, and by the way, again, I'm omitting the mathematical formulations in the interest of time, but for those of you who are interested, I should say that we're using beta distributions to estimate probabilities of outcomes. And then from those beta distributions, we can infer expected values and uncertainty, and we're using a similar functional form for novelty. But by the end of the talk, if people are more interested in this, I'm glad to discuss it in more depth. So after defining all of these computational variables, presumably some integration process needs to occur so that we can convert these all of these separate variables into some integrated utility for each restaurant so that we can compare these variables in order to make a decision. So in the end, our goal will be to find some function f which can map decision variables for each restaurant into the probability of choosing each restaurant. And then after that, our goal will be to map elements of this function to brain activity. So here's what we want to find out, right? Here are our hypotheses. Can we find a way to integrate decision variables, which is compatible with the behavior that we observe in patients? Secondly, if so, can we map how these decision variables and their integration are represented in the human neurons? And thirdly, can we map how decisions are being represented neurally? With these objectives in mind, we gave patients an exploration task. So here's where I'm going to talk about the game that the patients actually played. This task consisted in binary forced choice trials where patients would see two slot machines. So this is what patients would see on the screen in every trial. Patients would then choose to play one of the slot machines, which would then get highlighted like this. And then a binary result would get displayed on the screen, which could be a win or a no win. So that's it. Patients would get a binary result. Each one of these slot machines would have a fixed probability of dispensing a reward to the patient. And patients needed to learn which slot machines were rigged in their favor or against them on their own. So that's something they had to learn to maximize their rewards. After this outcome period here, where we're showing the result, the trial would just end, and then a new binary choice would be presented on the screen, like this. So notice here how this new trial includes one familiar slot machine and one entirely novel slot machine. So this design allowed us to have exploration of novel stimuli, as well as exploitation of stimuli that are well known by the patient or familiar to the patient. To help patients memorize and identify these slot machines, we added these paintings in front of each one of the slot machines. And I should say as well that in every trial, a pair of possible slot machines was chosen from a set of 3 possible slot machines that was, and these 3 were constant for each block of trials. This is the general timeline for each trial the patients performed. And let me talk for a second here about task structure so we can all be on the same page. I'll just mention the general structure briefly. First of all, patients performed in 20 blocks of 15 trials. And this block structure was what allowed us to disentangle novelty from uncertainty. So this is why I want to mention this a little bit. First of all, we instructed the patients that reward probabilities for each slot machine would be reset from one block to the next. So this is an important detail. So that's why I'm going into more detail here. So this reset from each block to the next meant that stimuli in each new block would be maximally uncertain again. So uncertainty would be reset. But novelty, importantly, wouldn't be reset because some of those slot machines had already appeared in the previous blocks. So in every new block, uncertainty was maximal again, but novelty was not because some of those slot machines were familiar. Just to make this very clear. To ensure that some of these stimuli are still novel on a block by block basis, in every block, we still went there and replaced one of the slot machines with a new one every block. So in every block, there would be 2 slot machines that were familiar and one of them would be novel. Okay, so now that I talked about task structure and about our objectives here, I'm gonna delve into behavioral data. So after we gave patients this task, what happened to their decisions? Did these variables of interest correlate with decisions in any way? So to measure that, I performed a logistic regression. First of all, a pretty simple analysis, the objective of mapping the effects of decision variables over patients' decisions, just to see if they correlate. So decision variables here consisted in expected values, which I'm defining here, which I'm indicating here by the letter Q, uncertainty and novelty, and their respective interactions with trial number within a block. So here in this equation, what I'm asking is, what is the probability of choosing the left option of the screen, for example, given the difference between each one of these values for the left and the right option? Can I map these correlations with this logistic regression? So here's the result. In this plot, every dot is a fit for one patient. So for these logistic regression coefficients, what I want to do is infer which factors were individually correlated with decisions. Here, the y-axis reflects whether patients sought or avoided each variable. Crucially, here, we found that patients learned to seek out options that had been rewarding in the past, which is reflected here by how their decisions correlated with expected values. So this pattern here means that patients were in the seeking region, meaning that they learned to seek options that had been rewarding in the past. So this is good. They're learning to optimize their decisions. But importantly, one effect that I want to talk about here is that the behavioral effect of uncertainty and novelty on behavior was dissociable. So specifically, while novel options were generally sought after, patients would click on the novel option more, uncertainty tended to have a more diverse effect on behavior. Some patients tended to seek it, while some patients tended to avoid it. So given this, we propose an adapted reinforcement learning model to explain patients' decisions in the task. We tested some different mechanisms, and we found that this one here that I'm going to talk about performed better in predicting patients' decisions, both actually in our patient cohort, but also in a cohort of neurotypical subjects performing in an equivalent task. So let me talk about this model here. This is just a schematic. I'm not going to show all the equations of the model, but I hope that this schematic can illustrate the important elements of the model. So the way the model works here, we're trying to predict patients' decisions. So to do that, we need to estimate choice probabilities. So in every trial, I have an estimate of how likely each choice was, choosing the left option or choosing the right option. So to do that, I derive these choice probabilities from my utility value that is assigned to each slot machine. So this utility value is constructed by adding expected values with an uncertainty bonus. So this is the key contribution of this model here. This uncertainty bonus reflects the tendency that each patient has to explore uncertain options. The way we're constructing this uncertainty bonus is by having novelty and uncertainty interact in a nonlinear way. I can show details later, but I should just mention here that we are doing a nonlinear interaction between novelty and uncertainty. They go and create this uncertainty bonus. This is in turn added to expected values to create utilities. What's important here is that this structure allowed us to capture individualized uncertainty preferences for each patient. So this number here, this uncertainty bonus, can be either a smaller or a larger number, depending on how uncertainty seeking a patient was. So this is the model that we're using. And just to summarize what we have for behavior, right? We saw that patients learn to make appropriate decisions as a task, which reflected the past history of rewards with the dissociable uncertainty and novelty behavioral effect. And additionally, our model suggested a mechanism where uncertainty and novelty can interact in order to drive decisions. So now we can turn our attention to neural data, which we recorded in this study. We're going to focus on pre-supplementary motor area, anterior cingulate cortex or dorsal anterior cingulate, and ventromedial prefrontal cortex. So this is the number of neurons we collected across 20 patients. Our overarching objective here will be to map out how these computational variables that I was talking about correlate with activity in these neurons, right? So to do that, I performed a forward encoding analysis with Poisson GLMs. One model that I'm going to focus on right now, and this is why I'm showing it here, is the encoding of positional variables. So by positional variables here, I mean separating the variables that pertain to the left slot machine on the screen and the right slot machine on the screen. So I'm separating here expected values, uncertainty, and novelty specifically for the slot machine on the left and the slot machine on the right of the screen. And I'm treating them separately in the model. Additionally, to make this work, we needed to correlate spike counts to each one of these variables in some time period. So I chose these time periods here specifically, either at trial onset, after we first display the slot machine on the screen, or right before the patient performed a button press. So we counted spikes in those time windows, and then we correlated spike counts to the variables of interest. So let me show you here one example of a neuron that does correlate with expected values. And so let me explain what's going on here. This is one example of a pre-SMA neuron, pre-supplementary motor area neuron, whose response is tuned to expected values for the slot machine presented on the right side of the screen. On the top here, we have a spike plot where I'm showing every trial. So every row here is a trial, and every dot indicates an action potential from this neuron, while this black line here indicates response times by the patient. So what we're seeing here is, and by the way, in the bottom here, we have average firing rates across time, and we have in each different color the level of expected value in each trial, right? So what we can see here is that this neuron tended to fire more when the patient saw a slot machine with a low expected value on the right side of the screen. And whenever expected value varied on the left side of the screen, this neuron just didn't vary as a function. So this is one example, right? But we can find many neurons, both in pre-SMA and other brain areas that do this. And in fact, if we map this across all regions and all time periods, what we see is this. So what I'm showing to you here is the percentage of neurons which significantly encoded each one of these, the percentage of neurons that encoded expected values positionally for each one of the brain areas. So for example, we observe here that positional expected value neurons were at a significant count of neurons in VMPFC and pre-SMA. And in pre-SMA specifically, we saw that in both time periods, we had a significant number of neurons that responded to expected values. So to make it very clear, the more that the patient thought they were going to get a reward as estimated by our model, the more these neurons were firing in these time periods. So it could be the other way around. It could be that a neuron was correlated inversely. It could be that a neuron was correlated in such a way that the less that the patient thought they were getting a reward, the more the neuron fired. So we could have neurons that correlate either way. So I'm just showing correlations here. So this is a similar pattern that we observe for all variables of interest. And at trial onset, expected values, uncertainty, and novelty were all being encoded in pre-SMA, while VMPFC tended to encode these variables in the pre-decision window. So note that novelty here was also being encoded by VMPFC in the trial onset period. So this here means that we found evidence for the components of value being encoded in VMPFC and pre-SMA separately for each slot machine. But now what we want to talk about is, how about their integration into some utility value that serves as input to decisions? So we found that utility was preferentially coded in the pre-SMA. Importantly, we found that a significant proportion of pre-SMA value neurons had their activity better explained by an integrated utility value rather than just encoding individual utility components. So this is really important, and we confirmed this result with the populational decoding analysis. I'm not going to go into details here, but we're running we're just creating a pseudo population of neurons across patients, and we're running decoding across all neurons. And we do find a similar pattern where decoding of utilities is more prominent in pre-SMA. With this in mind, we can now ask how decisions themselves are being encoded. And our key finding here was that pre-SMA was also the only brain region that we found that encoded which decision was made by the patient, encoding whether the patient pressed the left button or the right button. And this is one example neuron that we saw for this. The same was true for population decoding. And note here how decodability for decisions tends to peak right before the button press was executed by the patient. To further understand how pre-SMA neurons were encoding values and decisions, we performed a dimensionality reduction analysis, and then we mapped when in time representations for different decisions and different values, values, I should say, were most separable. Okay, so to make this very clear, first of all, we separated trials into low utility or high utility trials, or into left decision and right decision trials. So we created these different subsets of trials. Then, under this dimensionality reduction framework, we projected data, just the raw spike data, onto these principal components that we had for value or decision. And then we computed the Euclidean distance between projections for the trial subsets that we had separated. So this, in the end, what this allowed us to do was determine that while pre-SMA was encoding both values and decisions, low and high values were separable in the pre-SMA population activity earlier than decisions were. And this is what's reflected in these separability plots here. So one last result that I want to talk about here is, after a decision is computed by the brain or represented by neurons, what happens with the representation of the stimulus that was selected by the patient? So this is one example of a neuron in VMPFC that represents the expectation of values, but only for the stimulus that was selected in each trial. So that's contingent on selection. In fact, we ended up observing a similar pattern for all variables of interest. The VMPFC was the area that preferentially encoded these variables that were contingent on the decision that was made on each trial for the selection stimulus. So this concludes the results for part one. So just to summarize everything here, these findings that we have suggest that these value-based decisions during the explore-exploit trade-off depend on these highly specialized computations that are carried out in specific areas of prefrontal cortex. Additionally, the existence of an integrated utility signal at the level of signal neurons that we found suggests that these relevant decision variables, such as expected value, uncertainty, and novelty, are being probably integrated into a utility signal, into a unified neural response prior to being entered into a decision comparison. And I hope that this expands our understanding of how utility-based choices are being implemented in the human brain. So this was the end of part one and our study about decision-making under uncertainty. There's a lot of terms that are already introduced. So part one was a little bit longer because I introduced a lot of terms that I'm not going to reintroduce again for these other parts. And let me move to the second part, the second study that we did. So here, I would like for us to take a little bit of a step back and examine one of the most fundamental forms of learning, that is Pavlovian conditioning. So in our first part, we were more concerned with decision making, and here in the second part, and as well as in the third part, we're not going to focus on the decision parts as much. Instead, we're going to take a deeper look at how value learning itself is performed. So Pavlovian conditioning here specifically will be examined under the lens of cognitive maps being formed in the human brain. So this is a study currently in preparation, and we're doing this in collaboration with Christos Korelis, who is another PhD student at Yuli's lab. So let me talk about Pavlovian conditioning a little bit, just for background. The most basic form of Pavlovian conditioning is it works by associating some stimulus, which could be a sound tone or some image, to a reward that can be a piece of food. After learning the association, just being exposed to the stimulus can now generate some anticipation response, such as salivating, indicating that a food reward is expected. Now, Pavlovian conditioning can actually take on more complex forms, and I'm going to illustrate this with the issue of sensory preconditioning. So imagine that a dog is learning to associate two tones, A and B. So the dog learns that A and B are two tones that are played together. Now, at some point, tone B starts to be presented together with food until the dog learns that there's a new association here. Now, I have a question for you to ponder. What do you think should happen if the dog is now presented with tone A, which has never been presented together with the food before, but was associated with tone B? So, as you might have guessed, presenting tone A will elicit a salivation response in many cases, even though tone A was never presented in conjunction with the food before. So this phenomenon was first described in the 40s and was named sensory preconditioning. And I want to draw your attention here to a crucial part of this learning process, which is the association between tone A and tone B. As it turns out, what made it possible for the dog to predict that tone A was somehow related to the reward as well was that it learned an association between tone A and tone B. So as it turns out, many models that have historically been used in explaining Pavlovian conditioning, these models do not incorporate any explicit knowledge of the transition structure between different states of the world. And therefore, these models historically have struggled in explaining phenomena such as sensory preconditioning. So one modeling framework that lends itself really well to understanding how stimulus-stimulus associations, and more generally, to understanding how cognitive maps between states of the world are formed, this framework is model-based reinforcement learning. Learning in these models typically occurs by experiencing a surprise signal whenever you land in a state of the world that you did not expect. To study this specific phenomenon, we're going to investigate in this study here model-based Pavlovian learning in human neurons, utilizing a hierarchical conditioning paradigm that we created for this purpose. So to understand the formation of cognitive maps during Pavlovian conditioning, we set out to answer a number of questions here. First of all, can we find neural evidence for the encoding of stimulus-stimulus associations? We're particularly interested in the VMPFC, which past literature has shown to be involved in various forms of value-based learning, along with orbital frontal cortex and amygdala. Also, can we find evidence for the role of amygdala itself in this process? Previous studies in rodents and monkeys have highlighted how the amygdala is involved in forming estimates of expected values, for example, during Pavlovian conditioning. And finally, can we find evidence for the learning signal itself, which triggers Pavlovian learning? So for this, I use this two-step Pavlovian task. What patients would see would be this. They would first see a fractal image on the screen, which we name distal because it's far away from the outcome, followed by a second image, which we name proximal because it's close to the outcome. This stimulus would then be followed by an outcome, which could be a video of my hand putting candy in a bag, symbolizing reward, or my empty hand approaching the bag, which symbolized no reward. Patients were instructed, and this is very important, patients were instructed that every time they saw the candy video on the screen would contribute to a total amount of real candy that they would receive at the end of the experiment. So I actually brought in candy to the patients, some candy of their choice. We had a total of 4 trial types in this task, which I'm going to describe quickly here. So to help us understand how stimulus-stimulus associations were being formed, our task had a probabilistic transition structure in which 2 stimuli, A and X, let's say A and X, typically led to a reward most of the time, while 2 stimuli, let's call them B and Y, typically led to no reward. And in rare transition trials, however, the distal cue would lead to a flipped proximal cue, which then would lead to its usual result. So notice how this A, most of the time would mean they would get a reward, but in the rare transition trials, seeing A would mean that you didn't get a reward. And the proximal cues X and Y were always deterministic, that you would get a reward or not. This is a very simple probabilistic transition structure. And this is a diagram here just to illustrate. This is a diagram of all the possible transitions in our task structure. We propose a normative model-based learning algorithm for how patients are performing this task. And in this normative model, patients were constructing a transition matrix for the transitions between each one of these states into the other states of the task. So for instance, let me explain this. Let's say here we have the probability PAX, which is the probability of transitioning from the distal QA to the proximal QX. So the true value, the ground truth for this number that I hard-coded in the task is 75%. If you're in A, there's a 75% chance that you'll transition into X. However, patients didn't know this, and they had to learn these probabilities during the game. So we created an entire transition probability matrix containing each one of these p-values. And this matrix represents, in fact, our best estimate for the patient's cognitive map for the structure of this task. So for the patient's cognitive map, we could now estimate the transition probabilities to infer certain variables, such as, for example, the expected value of reward in each trial. And by the way, I should mention here that X and Y were reversed at each block. So that's why you see these patterns of going up and then down. So we introduced reversals just to make sure that there was continuous learning. So this is expected values for X and Y, and they would keep flipping like this. And another thing we could estimate from these models was the amount of surprise that was caused by landing in a given state, which we're calling here state prediction error. Landing in an unexpected state, that would trigger high amounts of state prediction error. And one of our hypotheses here is that this is the very signal that's being used by the brain to update cognitive maps. Additionally, at the time the first stimulus is presented, we can already infer from our cognitive map which stimulus is more likely to be the second cue in the two-step task. So this is really important because if we find evidence that the identity of the second stimulus is already represented neurally at the time of the first stimulus, we can interpret this evidence of the brain encoding a stimulus-stimulus association, which we think is an element in the construction of cognitive maps. So given this whole framework, what does the patient data say? So first of all, subjective preference ratings for all fractal images were obtained. And we saw that in between blocks, patients we asked patients to re-rate the fractals that were included in the previous block just to measure changes in subjective preference. And when we group distal and proximal fractals together, we observed that the more commonly rewarded stimuli were rated significantly higher than the stimuli that were only rewarded in rare transitions. Additionally, one behavioral effect that I should mention here is that we found that there was an interaction effect of expected values and prediction errors in pupil diameter. So we also got eye tracking from patients. And this, I believe, adds further behavioral evidence that some form of learning and conditioning here occurred during the task. So let's now take a look at the neural data that we obtained from a total of 13 sessions with these patients. So first of all, and this is a key result, the VMPFC was the only brain area which we found to significantly encode the presumed identity of future stimuli. In this plot, I'm showing here the decoding accuracy on a patient-by-patient basis, so each dot here is a patient. This result means that the VMPFC successfully encoded stimulus-stimulus associations which could be utilized in the construction of cognitive maps. Additionally, the VMPFC and amygdala encoded expected values as predicted by the model-based learning algorithm, which is consistent with literature in the role of these regions, right? And one more thing that I should mention here is anterior cingulate and pre-SMA robustly encoded whether the patient received candy in a trial or not. On top of that, these brain areas also encoded the extent to which patients were surprised by the result of a trial as predicted by the model-based algorithm. So this concludes part 2. So just to put it briefly, these results position VMPFC in a very interesting spot in model-based learning during Pavlovian conditioning. And hopefully this expands our understanding of how Pavlovian conditioning occurs in the context of encoding cognitive maps in the human brain. So these are our main results on creating cognitive maps during Pavlovian conditioning. So now with the time that I have left, I want to talk a bit about our third part, which is a study that we published in 2020, which is about observational learning. So now that we talked about decisions under uncertainty, and now that we talked about Pavlovian conditioning, we can investigate a specific application in social cognition. So I ran this study in collaboration with Yuri Minsha, who is a former PhD student in Yuli's lab. And let me just give a very quick background here. So learning about rewards can be done either by experiencing rewards yourself or by observing somebody else get rewards. And one form of observational learning is vicarious observational learning, right? Which is very simply put, you just learn about the future rewards you can get by watching somebody else do it, right? By somebody else get rewards. So the amygdala was our key region of interest here for several reasons. There's a wealth of studies in the past that position the amygdala in a very key spot in encoding values and encoding observational learning, both for emotions and for value-based learning. So for this reason, in this study specifically, we just wanted to look at the role of amygdala in observational learning. And specifically, what we want to find out here is, first of all, we want to discuss and see if we can map which behavioral mechanism is being used to perform observational learning. And on top of that, we want to map out how value learning is being carried out by amygdala neurons. We want to see if value-based variables correlate with activity in amygdala neurons. And can we distinguish observational learning from self-experience learning in these neurons? So to do this, we created a very simple Pavlovian slot machine task. So this task consisted of 288 trials that were distributed in self-experience and observational blocks. In the experiential block, patients would see one of two possible slot machines, and any rewards that the patient won here would be added to the patient's own reward total. The slot machine would just play automatically and then dispense a reward according to some underlying Gaussian distribution that we created. In each block, one of the slot machines would be good and one of the slot machines would be bad. Patients needed to learn which one was which just by paying attention to which rewards were being dispensed. In the observed blocks, patients instead observe a video of another person playing the game. And importantly, they do not accrue the rewards that the other person got. And just to make it clear, we also added another subset of trials in which patients were tested in their knowledge of the game. So they would see two slot machines and they had to choose one to play. We use those trials just to infer patients' estimates of the expected values from each slot machine and to fit computational models of observational learning. So we compared, without going into too much detail, we compared different reinforcement learning mechanisms, and we found that a model with a single learning rate performed better across self-experience and observed learning. We also found that the winning model incorporated a counterfactual learning mechanism in which the value of the unseen slot machine was updated in the opposite direction to the slot machine the patient saw. And one behavioral effect that I'm going to mention here is that the percentage of choices where subjects picked the best decision was equal across self-experience and observed trials, which means that patients were learning in both cases. So we recorded 202 amygdala neurons from these patients, over 12 people. And then we performed some decoding analysis. So first of all, we saw that amygdala strongly encoded whether a trial was observational or self-experience. In this plot, the red line here indicates actual decoding accuracy, while the histogram indicates the null distribution using shuffle data. Additionally, we observed that amygdala neuron populations strongly encoded the amount of outcome that was received in a trial, both by oneself and also the outcome that was received by the other agent. Another thing we found was that amygdala neurons at the level of populations also revealed a representation for how expected values were being encoded in the self-experience case. And this is one example of a neuron that encodes expectations for value in these self-experience blocks. So see here how if there was a low expected value, this amygdala neuron tended to fire more in those trials. So, however, despite being unable to get a significant populational decoding for observational expected values, we did find a significant neuron count individually for both types of expected value. And this discrepancy between the analysis, we accounted for it by noisy weakly coding neurons, which we explain away in the study using a simulation part, which I'm not going to include in this presentation, but whatever, we can talk about it later, or it's present in the study. And the last thing I want to mention here is we also asked whether the encoding of outcomes and expected values occurred in the same subsets of neurons across self-experience learning and observational learning. In the x-axis here, we have sensitivity during self-experience trials, and then in the y-axis here, we have sensitivity during observational trials. As it turns out, both for expected values and outcomes, the self-experience and observational representations of value occur almost completely in disjointed populations of amygdala neurons, meaning that we couldn't find a generalized code for value in this region. So to conclude part 3, we tracked how value-related variables were encoded in amygdala during observational learning. And we saw that self-experience learning and observational learning in amygdala were encoded differently, not only differently, but by separate neural subpopulations. And on top of that, we proposed a counterfactual computational mechanisms that explain patients' decisions in the task. So this concludes part 3, and this concludes, in fact, the scientific results of my thesis. So overall, I hope that I convinced you that we mapped several aspects of how value-based learning and decision-making are being encoded by individual neurons in the human brain. I hope that this work also highlights the value of considering behavior front and center along with the neural data in the effort to understand human cognition. So I hope that this has become really clear. And one more thing I want to mention here in terms of future work that can be done in this field is I think that my work can be understood as a basic science stepping stone in a direction that can aid some endeavors such as computational psychiatry, for example. So the field of computational psychiatry is engaged in applying these same computational descriptions that I'm talking about in assisting in finding biomarkers for mental illness or potential treatment routes for disorders such as OCD, schizophrenia, and depression. And I hope that this work can pave the way, at least in terms of mapping the computational constructs in the human brain. And one more thing I want to mention here is the integration of neuroscience and reinforcement learning is always evolving, and it has been very successful for many years. So I'm really excited about new developments like deep RL and distributional RL, just to cite a few. And I think they will start to have a growing impact in our understanding of brain function. And it's a very natural direction for this work to progress in.